bit about antibody structure and where a lot of the diversity in antibodies comes from. I'll say a little bit about monoclonals and the different types of monoclonals. I'm not going to be saying very much about their therapeutic use because Mike Tomlinson knows a lot more about it than I do and will tell you a lot more about it than I can. So just to remind you, I'm sure all of you have been vaccinated against a variety of interesting conditions. Vaccination is essentially just pre-exposing you, i.e. your immune system, to something that boosts the immune system, enables it to recognise that something without actually causing the disease itself. And I gave you this list last time of all the different things, or some of the different things, I should say, for which vaccines are now available, of which the most spectacular really is smallpox, because smallpox has essentially been eliminated as a disease worldwide, and with polio we're pretty close to eliminating that as well. So vaccination programs, if carried out with fully active vaccines and with very well organised systems for distribution and vaccination, can actually reduce the load of some of these infectious diseases. Of course, having a fully active vaccine is a very big challenge, and having a successful method for disseminating it and getting it used worldwide is a much, much bigger challenge. So it's both a scientific medical challenge and an enormous logistic and to some extent political one. But there are plenty of diseases for which there aren't decent vaccines available, or where the vaccines that are available give rather poor protection, maybe 30-40% or so. And some of these are major killers. HIV, obviously, malaria, diarrheal diseases, TB, still account for very, very significant numbers of deaths worldwide. And there aren't, with the partial exception of TB, there aren't good vaccines against these. And the TB vaccine is by no means perfect. And it's often the case, actually, one of the problems with vaccine development is a lot of it is done in Europe and the States, but different populations around the world respond very differently to vaccines. So there's no guarantee at all that a vaccine that works well on a predominantly Caucasian population, for example, will be successful or as successful when it's used on an Asian population. And then there's huge problems about things like storage. It's one thing to develop a vaccine in a place where you've got access to fridges and -20 freezers. It's something else to get that vaccine out to a place where the nearest fridge might be 100 miles away and where the vaccine might be quite unstable. So vaccine development is a big problem. It's also an issue, of course, economically speaking, because the economic return on vaccines isn't that great. They're used once and never again. Drug companies like to sell something that's going to cure you in the short term, but which you're going to need again in 6 months' time. So there are rather few commercial entities prepared to put the time and energy and finance into developing vaccines. They tend to go, vaccine development is done much more at the sort of pure research level, funded by organisations like Wellcome Trust and Bill and Melinda Gates Foundation and so on. As I said, these are big killers. I showed you these figures last time. So a lot of people die from these diseases every year, have very significant economic impact. So the basic principles of vaccination are pretty obvious. First of all, the vaccine has got to be safe. There's no point in giving people a vaccine against an illness if it just makes them ill with something else. That's not to say that they don't have side effects. I'm sure all of you at some point have had a vaccination that's made you feel like you've had flu for a couple of days. That's just your immune system doing what it's supposed to do. You might get a very stiff arm. There are some examples of vaccines in early use, of course, which were quite dangerous and where there was a risk of delivering the disease that the vaccine was supposed to be protecting against. I'll say a bit more about that in just a moment. So you want your vaccine to be safe. You want the vaccine to be as long-term as possible. So you're hoping once you've administered the vaccine that the person's immune response is going to be good enough to cope with that disease for a significant period. There's not a great deal of point in having to revaccinate every 6 months. There's some features about the biology as well. So, for example, if you're dealing with a pathogen that targets a particular cell type and that cell type is very important, then you don't want your vaccine to cause any damage to the cell type. The vaccine's got to be a neutralizing vaccine that destroys the agent that's causing the infection and not the infected cell. A good example of this is polio, which infects neural cells, nerve cells. So if your vaccine destroys polio-infected cells, the effect is going to be pretty much the same as the polio virus itself. You want a vaccine that's capable of recognising and neutralising the virus before it spreads through the body. And obviously, for economic reasons, the things I've just talked about, you want your vaccine to be as cheap as possible. You've not got rich people paying for vaccines by and large, and you want it to store well because you want it to be able to be kept on the shelf in a doctor's surgery or a dispensing chemist or a nurse's store under a wide range of conditions, maybe very hot conditions sometimes, and not to go off, not to lose its potency. So this is your ideal vaccine. Of course, a lot of vaccines don't measure up to that. Now, biotech and the use of cloning methods has led to some significant developments in vaccine safety, and I'm just going to highlight a couple of those briefly before we go back to thinking about antibodies. So just thinking a little bit about what the vaccine actually is. The original vaccine, the first ever vaccine, as far as we know, the cowpox vaccine that I discussed last time, was essentially a live virus. And that virus resembled the smallpox virus, which it was vaccinating against. And the principle there was simply it's better to have cowpox, which is a relatively mild disease, and gives you long-term immunity against the smallpox virus than it is to have smallpox. But often, as vaccines became more developed, and obviously we've moved on a great deal since the days that we were just infecting people with cowpox, there are different ways that have been used to develop vaccines. And a common one is to take a pathogen and to attenuate it. And what that means is typically you'd grow the pathogen like it might be a virus, repeatedly under tissue culture conditions. And as you do that, its potency, its ability to cause infection in the original host goes down, and that's called attenuation. Obviously, you hope that its ability to provoke an immune response doesn't go down. So what you're looking for is a pathogen that's no longer capable of causing a disease or causes a disease much less effectively, but can still provoke an immune response, can still be seen by your immune system. Now, classically, this was done by repeated serial passage. So viruses would be infected into things like eggs, for example, and then harvested and then reinfected, and they would gradually lose their potency. Of course, once you can start to manipulate an organism, once you can use recombinant techniques to go in and make mutants in that organism, you can do things like actually take out the bits of the genes that are responsible for the organism's ability to cause disease while not removing the genes that enable it to provoke a response from the immune system. So you might, for example, be blocking, removing genes that encode a toxin, to take a simple example, but you're not going to change anything else. And these are called live attenuated vaccines, where the vaccine is still alive, it's still a fully functional organism, but it's had its ability to cause the disease taken away from it. There are some issues with this. There's a lot of horizontal gene transfer that takes place between organisms, and so there might be a concern that your attenuated organism could reacquire the bad genes, the genes for the toxin or the genes for some other aspects of its ability to cause disease. Nonetheless, live attenuated vaccines are one of the things that people work on quite a lot. Obviously, you can also use killed cells, so you take the pathogen, you kill it in some way, heat, radiation, that sort of thing, and then it's no longer capable, of course, of causing a disease, but again, as long as the antigens on the pathogen are present, it should still stimulate an immune response. Now, both of these first two things come with a certain amount of risk. The attenuation may not be sufficient. The organism may reacquire genes that you've removed from it. And if you kill the cells, you've got to be sure that they're all dead. If there's some living cells left in your preparation, then you may be injecting somebody with live organisms or live virus that can cause the disease that you're trying to vaccinate against. So a much better method, ideally, and again this is to a certain extent in theory, is to purify away or have a limited number of components from the pathogen that are able to cause an immune response but completely incapable. There's no circumstances where they could cause disease. And these sorts of vaccines are becoming much more common. Quite often vaccines are injected with something else, and that something else is actually often quite a crude mixture of things, but it has the property of giving the immune system a bit of a kick, stimulating the immune system so that it's better at recognising the antigen that you're injecting. And there's something else is referred to as an adjuvant. And adjuvants themselves have their own controversies around them and their own issues around them. I'm not going to say anything more about them now, but if you hear the word adjuvant referred to, that's what it is. It's something that the vaccine is, well, the purified antigen is mixed with in order to provoke a stronger immune response. I'm not going to talk about the mechanism of that now. In fact, the mechanisms aren't entirely clear. So if we think about applying biotech-type approaches to vaccine development, the obvious one would be to look for the proteins which are expressed by the cell or which are present on the virus that cause the biggest immune response, and then just express these proteins on their own and use these proteins to do the immunization. And it might not even be a whole protein, it might be a part of the protein. People will refer to subunit vaccines, which are vaccines which usually just express typically a viral coat protein, for example, or part of a viral coat protein. Here's a really classic example. This was work that was done back in the 80s on the hepatitis B vaccine. And this is the first case where someone had shown that you could take a gene for a protein that was made by an infectious organism, in this case, the Hep B virus. You could express it in bacteria, and you can make the protein, and then use that to immunize and show that the organisms, in this case, monkeys that had been, chimpanzees that had been vaccinated with this purified protein, were then resistant to being infected by HBV. So this was proof of principle, essentially, that this kind of approach would work. But actually, some of the people involved in this, including Ken Murray, went on to be very involved with developing the vaccine that was subsequently the one that became marketed against HBV. I don't know if you've noticed, by the way, but the teaching block that we often have our lectures in, the first building you pass on campus as you come out of the station on your left-hand side, is called the Murray Building, and it's actually named after Ken Murray and his wife Noreen Murray, who were both students at Birmingham, met here, and made a very large amount of money out of the vaccine development and donated a lot of it to foundations to support overseas students coming to study in Birmingham at PhD level. So when you walk into the Murray Building, although I don't think there's anything on the building that says this, it's essentially celebrating the success of Ken and Noreen Murray at developing these techniques. This sort of thing would be pretty routine now because the kits are available and lots of vaccine development has been done. Of course, in those days, they had to do everything from scratch. So they had to work out how to do the cloning and develop the appropriate vectors and so forth. So it was a real tour de force. Here's a picture of the HBV, two different diagrammatic representations. This is the whole surface of the virus. This protein is referred to as surface antigen because this is the thing that your immune system sees first of all. There's also, it's actually an example of a virus where there's an outer coat and then an inner coat, and then the genome is inside that. So there's also what's called the inner core here. And either the surface antigen or the core antigen can be used to immunize monkeys and indeed people against HBV infection. So once you've got the immune system recognizing those antigens, you're then essentially resistant to infection by the virus because the antibodies will neutralize the virus if it gets into the body before it can infect cells. Now, the great advantage, of course, of this kind of vaccine is there's no risk of infection. You're not using any genome DNA, you're just using a part of the virus that provokes the immune response, but there's no information, there's no genetic information about the infection going in that injection whatsoever. So this is clearly better than using an attenuated virus or a virus that's been killed in some way, if you can kill a virus, because the risk of infection is zero. So this is why this kind of approach to vaccines has become much more popular and much more widespread, and quite a few vaccines now as subunit vaccines. The trick, of course, is to identify the protein that is going to give you good immunity, and that's relatively straightforward with viruses because they don't have very many proteins. It's much trickier with other organisms. So two companies were involved in this, Biogen, which was based in Switzerland, and Chiron, that was based in California, has now been taken over by Novartis. And it was Biogen, in fact, that Ken and Noreen, Ken in particular, did a lot of work for, and That's where they made their fortune. They were a great couple. They're both dead now, Ken and Noreen, but they used to come to Birmingham on a very regular basis and visit us and check out how the students were doing. They were multimillionaires. They'd made a vast amount of money out of this. And they were the most delightful, quiet, modest people you could hope to meet. And I can remember once they came down and we took them out for a meal. And I knew that both of them were quite keen on gardening. And I said, do you fancy going? I can take you around the University Botanical Gardens tomorrow. This is the weekend. And Noreen said, no, I need to get back to the lab. So this was a Saturday afternoon. Her plan was to get on a train, go back up to Edinburgh, and be back in the lab on Sunday. And this is despite the fact she was in her 70s at the time and worth, £10 million or so. So I had a lot of respect for them. And a lot of us are very grateful to them because their funding has supported quite a few PhD students coming through Birmingham. It all began with the vaccine against HPV. As I just mentioned as well, you can also use the biotech kind of approach to engineer organs to attenuate them. And this can be particularly useful with more complex organisms where they have a complicated route of infection and you want to interfere with that in some way to block the infection and to provoke the immune response at the same time. And a classic example of this is malaria. Now, no one's got a decent malaria vaccine yet, but there's quite a lot of work being done on it because malaria is a big health problem in many parts of the world, both because of its high mortality rate, but also because it takes a lot of people out of being economically productive because they get this recurrent fever, which can lay them very low for long periods of time. And the malaria parasite's got a very complicated life cycle, but it hides out in the liver. And we'll go through this life cycle. Eventually, it will produce these things called myrizoites, which are one of the forms of the parasite. It goes through multiple forms as it infects. And these can kill, or they can certainly provoke a very nasty condition until your immune system gets on top of them. And then the parasite goes back into the liver, shuffles its immune coat around a bit, and then re-emerges weeks or months later. So what people have been looking at is making mutant forms of the malaria parasite that are blocked quite early on in this infectious cycle, and so can get into the liver cells and can begin to develop, but can't go right the way through the full cycle. And the hope is that these liver cells will then display malarial antigens that will be recognised, will provoke an immune response, and that will give some measure of protection against the malaria parasite. It's unlikely to be complete because of this ability the malaria parasite has to rearrange its surface coat so that it evades the immune system. But this is certainly one of the routes that's being looked at the moment. There's some slightly more far-fetched ideas out there, which I think are attractive, although they've not really been demonstrated to work yet. One example is, why not make an edible vaccine? Why not make a transgenic plant that expresses antigens and then that can be administered as food? So the fancy idea that people had was, how about using bananas for vaccination? Make a transgenic banana that happens to be expressing some kind of, say, a viral antigen. When you eat the banana, that viral antigen is recognized by the immune cells in your gut as being foreign. They can provoke an immune response. And the great beauty of this, of course, if it were to work, and it's kind of a fanciful idea, but people are working on it, is you don't have a storage problem because the banana is doing the storage for you, and you don't have an issue with all the paraphernalia that's required for injection and so forth, because all you've got to do is eat the thing. So it cuts out a lot of the complications, or potentially could cut out a lot of the complications around storage and delivery and the need for trained personnel and so forth. Nice idea. There's been some preliminary work showing that at least in principle this could work. I don't know whether anyone's working full-time on a major vaccine program, but it's an example of the sort of thing that can become possible when you can manipulate the genetic material of plants as well as bacteria. And then another idea that's gaining a lot of currency at the moment, and actually turns out to be quite successful, is the idea about using the gene for the antigen as the thing that you vaccinate with, not the antigen itself. So everything we've talked about so far is to do with either expressing the antigen or having the organism expressing the antigen. So the antigen is the protein that is going to cause the immune response. The idea here is you don't need that. If you just inject the gene, that gene may get taken up into cells. Those cells then may express the antigen, and that will then cause an immune response to take place. Now, I thought this was a pretty crazy idea because I thought the level at which cells would take up DNA would be way too low for it to work. But it turns out it does work. And people have done experiments on this and shown under controlled conditions that you can actually raise an immune response just by injecting the gene. And clearly that gene's got to be taken up, transcribed, translated, and expressed before the immune response is provoked. And to my surprise, this actually can work. So it's an area. An area to watch, I think. A lot of development taking place around this at the moment. Okay, that's all I'm going to say about vaccination. I'm going to go back now to talking about antibodies, and I'm going to say more about their structure, because I said in the last lecture that I wanted to begin to link the structural nature of antibodies with the way that they work, and talk in more detail about this issue. Where does all this diversity come from? You have vast numbers of antibodies, vast numbers of B cells, vast numbers of T cells, capable of recognizing all sorts of different structures. And the amount of genetic information for that is more than you've got space for in your genome. So where does the information for this variability come from? So just to remind you a little bit about the basic structure, and then we'll dive into the structure in a bit more detail. Antibodies are produced by plasma cells, which are differentiated B cells. We talked about that last time. The receptors expressed on B cells are quite like the antibodies that they eventually produce, but during the process of maturation, the specificity of the antibody gets better and better, and we'll talk about that in just a moment. There are two functional parts. There's a bit that recognizes the antigen, the foreign substance, and binds to it, and there's a bit that then interacts with the immune system, so that's the variable part and the constant part. And the constant part, and we're using the word constant in a slightly misleading sense. It's not completely constant, as we'll see. It does vary between different antibodies, but the constant part is more constant than the variable parts are. The constant part is the part that's recognized by things like the Fc receptors that we discussed last time, that are capable of recognizing the antibody, and thus the thing that the antibody is bound to, and doing things like internalizing it, phagocytosing it, breaking it down, and so on. Just to remind you again, we'll look at some different ways of showing the structure. Here's a classic IgG type antibody. Here's your light chain. Here's your heavy chain. Constant region is part of the heavy chain only. The light chain doesn't have a constant region. And you've got the variable regions at the top. And as we'll see, this is what happens in humans. There are some organisms in which this is different, which is interesting, but we're talking about human antibodies at the moment. The light chains have got essentially a variable and a constant domain. There are two different kinds of light chains called kappa and lambda. These are just defined by differences in sequence, but I'll come back to that in just a moment as we talk about diversity. Heavy chains have usually got four domains. Some have got five. They have one of which is variable. The rest of them are constant. And there are different types of heavy chain, and these different types of heavy chain give rise to different kinds of antibodies. So depending on which particular sort of heavy chain you've got, you'll have a different kind of antibody. And these are called IgA, IgD, IgE, IgG, and IgM. And when you see structures like this, we're almost always talking about IgGs. Some are different, so some form larger complexes. I'm not going to say anything about that, but you may well come across it in your extra reading. They have slightly different functions. They have difference in sequence, but the IgG form is the most common form in the body, and that's what we're going to be focusing on, and that's what things like monoclonals, by and large, are based on. So a little bit more detail. I'm going to show you a full structure in just a moment. Just to remind you that you have, in addition to the protein chain, there are disulfide bonds that hold the different chains together. There's also carbohydrates attached to the constant regions of the heavy chain. So antibodies, of course, are secreted proteins, which means when they're made inside cells, they pass through the endoplasmic reticulum and through the Golgi body. And as they enter into the Golgi body, carbohydrates are attached to parts of the the constant region of the heavy chain. These are glycans, and these are quite important in terms of what they actually do in their mechanism, although they don't have anything to do with the recognition. There are also disulfide bonds within the chains as well as between them. So interchain disulfide bonds are these bonds here that link different chains together. So there's disulfides that link the two heavy chains, and there's disulfides that link the heavy chains to the light chains. But there's also internal disulfides in all of them, which are partially responsible for the conformation that the peptide chain in the antibody takes up. So they have to be disulfide bonded. Just mentioned the glycans, and the reason I want to emphasize these is that the disulfide bonds and the glycans can be quite important. The disulfide bonds in particular are very important for determining the structure and the function of those antibodies. Now that's fine when they're made in our bodies because our cells have evolved to put in the correct disulfides and the correct glycans. But if you suddenly start expressing these molecules in bacteria, for example, bacteria do not, by and large, glycosylate proteins. So a protein that's glycosylated in a human cell, if it's expressed in a bacterial cell, will not be glycosylated. It won't have those sugar chains attached to it, and that may change its function. Similarly, although bacteria and yeast cells do form disulfide bonds, in bacterial cells the capacity to do that is somewhat limited. So if you want to persuade a bacterium to put disulfide bonds into a complex protein, there's often quite a bit of engineering that has to be done to the bacterial strain to make it do that. So this is where it becomes very important to think about what kind of organism or what kind of cell are you going to use to produce your molecule in. And I refer to this a little bit in the BioM 10 lectures. You'll hear more about it from Dr. Huber, that there's always a lot of things to be considered here. You know, bacteria are very cheap, very easy to grow in large volumes. They're easy to handle, but they don't make all the modifications to proteins that we might want to produce a functional protein, and that's important. Because we can engineer the genes for antibodies, we can, of course, make lots of different variants that don't occur in nature. And this is just a diagram of some of the different ones that people have produced. So here's a classic IgG antibody. It's color-coded so you can see the constant and the variable regions. But many people say, well, actually, if all we want to do is have an antibody that's going to be used diagnostically or in a Western blot or something, we don't need all the paraphernalia that's recognized by the immune system. We just need a protein that recognizes our antigen really well. So we might use a thing like this, which is a short chain antibody which just consists of the two variable regions which have been covalently linked by a little linker there. These are Fab fragments which have had the tail of the heavy chain clipped off, so they only express the variable in the constant region from the light and heavy chain. This is a Fab 2 fragment, which is like this, but where there's enough hinge region left that it can be disulfide bonded together. So these have been engineered and experimented on, and in some cases people want to produce full-length antibodies, in some cases they want to produce Fab fragments or diabodies, depending on what application those antibodies are going to be put to. Okay, so getting into a bit more detail about the structure and trying to understand how the structure affects the function of the antibody. This is a structure of a typical IgG molecule. And of course it doesn't look like this. I showed you, we went through this thing about structural cartoons in the lectures last week. This is just a cartoon showing the approximate position of the alpha carbon chain. And these here, of course, are beta strands. And what you can see from this is that antibodies are predominantly made up of beta strand, in fact, organized in beta sheets. So when you've got several beta strands together like that, that's referred to as a beta sheet. And then with, obviously, bits of peptide linking those together. So there's very little or no, really, alpha helical region within antibodies. They're largely made up of beta strands and beta sheets. If we zoom in to the individual domains, there's two things we can tell. First of all the domains in antibodies contain two beta sheets. And second of all, the domains are actually quite similar in structure. And this is presumably a relic of some evolutionary process whereby there's been gene duplication and then some divergence between the duplicated genes or parts of genes to give the more complicated antibody molecules we have today. So if we zoom in, and this is looking now just at a light chain structure, we've got the constant part of the light chain and the variable part of the light chain. And this is the N-terminus of the protein, this is the C-terminus of the protein. You can see, I hope, by the colour coding, the two different beta sheets. So here's the first domain. You've got one beta sheet here made up of these 4 strands, and one beta sheet here made up of these 3 strands. And there between them is the disulfide bond that holds those 2 beta sheets in the correct register. And then in the variable chain, it's slightly different. You've got 2 beta sheets again, one made up by those 4 strands there in red, and one made up by those 5 beta strands there in blue. Now, it looks a bit like a plate of spaghetti, this, and one of the ways that we'll often look at protein structure is a very, very diagrammatic form, where we ask the question, if you were to trace the peptide chain, if you started at one end and you followed that chain through the molecule, which bits of the molecule would that chain represent? So essentially what we're trying to do is start here and then follow the chain around, and you can see it gets a little bit complicated because you can start to lose yourself in the structure. I'm just tracing it around like this. But one of the things that I hope you can see as I do that is that the strands in the individual beta sheets are not adjacent to each other in the sequence necessarily. So the chain will flip between the 2 beta sheets, even though structurally they're integral components. So that's shown better if you flatten the molecule out like this. So this now loses a lot of structural information. It doesn't tell you where they're organised in space, but it enables you to see how the chain moves through the different through the different domains. So here's our variable domain again. Here's the N-terminal region. If we follow that chain, we've got one beta strand, another anti-parallel beta strand. Then the chain moves across to the other beta sheet. You've got a couple of strands there, little one there, and then it goes down to the first beta sheet, another couple of anti-parallel beta strands, back to the first sheet, and then out, and that will then come into the second domain here. So this is not uncommon in proteins, that large structures within a protein, like a beta sheet, for example, if you actually follow the peptide chain, the chain is not necessarily continuous. It may move to other parts of the protein and come back to finish up that beta sheet. And this is why these cartoons, I think, are very valuable, because it's very hard looking at a diagram like that to follow the course of the polypeptide chain, and it's downright impossible if you do it with things like van der Waals radii. But if you display it that way, you can begin to follow how the chain moves. And this shows a couple of things. One is the clear conservation between these 2 domains, even though this one's the variable domain, this one's the constant domain. You can see the basic structure, the topology, is the same. The way in which the strand moves from sheet to sheet is the same between both of them. The difference is, or one of the differences, is that there's a couple of extra strands in the variable domain, so you've got 5 strands rather than the 3 that you've got there. But the actual topology, so this goes here, then across to here, exactly the same here, goes here, then across to here. And of course, if these have evolved from the same starting point, that's what you'd expect. So once that fold has evolved, and this is often called an immunoglobulin fold, once that fold has evolved, the cell is just finding lots of different ways of using the same fold to do slightly different jobs. And this is also true, by the way, of the B-cell receptors. Okay, so that tells us a little bit about the organisation of the molecule in space. It doesn't tell us anything about how that molecule is able to recognise other molecules. So this is what I'm going to say a bit about now. One of the things that was pretty obvious fairly early on, once people started sequencing antibody genes and looking at them, was that you could take different parts of the antibody gene from genes for different antibodies, look at the different variable regions in those antibodies and line them up and ask the question, where does the variation occur? If you take the variable region from antibody A, antibody B, antibody C, and you compare those to each other, what regions are similar, what regions are variable? And this has been done with large numbers of different antibody sequences. And it turns out that some regions within the variable domain are extremely variable. And these are sometimes called hypervariable regions. And other regions within the variable domain are more or less constant. They don't change very much in sequence between different antibodies. And these are sometimes called the framework regions. So you can take all these sequences and look at them and ask whereabouts on those structures are those hypervariable regions. So here's one way of doing it. In this case, we've just taken the variable region from either a heavy chain or from a light chain. So we're either looking at this part of the antibody molecule or this part of the antibody molecule. And the vertical axis here represents the amount of variability. So for each individual amino acid, how many different amino acids do you find at that position when you compare different antibody sequences? And the horizontal axis is the residue number. So we're starting at residue number 1, line up 20 antibodies, or in this case actually probably several hundred antibodies, and say, how often is that an alanine, how often is it a glycine, and so forth. And it turns out that for these first 30 amino acids, there's some variability, as you can see, but actually not very much. And then suddenly it shoots up, and then it goes back down again, and then it shoots back up again and goes down again, and shoots up and goes down. And pretty much the same pattern is seen in the variable chain. So these are the hypervariable regions. The regions in red are the hypervariable regions, and these vary a lot between different antibody molecules. Now that's well and good, but it still doesn't tell you whereabouts on the structure that variability is. But we can map that. We can take that diagram, that graph, and we can say, as we move along the structure, the three-dimensional structure, whereabouts are those variable regions? Whereabouts on the structure are they? And it turns out that they are in the regions that connect the beta strands and the beta sheets. So if we show it this way around, so again we're going back to this diagram now, here's the two beta sheets in the light chain variable domain, and we've got three hypervariable regions, hypervariable region 1, 2, and 3, and these actually map very precisely onto these bits of the protein chain that link adjacent beta strands or link the adjacent beta sheets. So that's where most of the variability is occurring. And if we now superimpose that onto the actual structure, we can now see, very strikingly, that actually these are very close together in physical space. So even though these are quite separated on the peptide chain, when that chain folds up, the variable regions are quite close together. And essentially what it gives you is a tip to this variable domain, which is highly variable. And the rest of the thing, although it varies a little bit, is providing more of a framework for that variability to be exposed. And I've gone into that in a bit of detail because it illustrates very importantly that if you want to understand protein function, it really helps to have a three-dimensional structure. If you didn't have that three-dimensional structure, you wouldn't know that these variable regions actually are physically in the protein all at the same position or very close together. So without the information about the protein structure, you don't have a complete picture. Once you've got the protein structure, you can begin to make some hypotheses about how this protein might actually be working based on the genetic analysis, which has told you where the variable regions actually are. Now, these regions are also called CDRs, or complementarity determining regions. Complementarity simply refers to two things that fit together. So the hypervariable regions are the regions which give antibodies most of their ability to bind lots and lots of different structures. You'll see them referred to as CDRs, and I'll use that terminology in just a moment. And the high degree of variability of these CDRs from one antibody to the next is largely, but not entirely, responsible for the huge diversity of antibodies that exist within the body. So the fact that we have the ability to recognize loads and loads and loads of different structures, including things that our bodies have never seen before, depends to a large extent on the hypervariability of the CDRs. The body can produce many, 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 many different structures in antibodies, and among those millions or billions of different structures, if you present it with some kind of foreign structure, one or more of those structures will recognize that foreign structure. It's quite remarkable when you think about it, because you think about all the different conformations that different proteins can take up, but you can pretty much guarantee that whatever structure protein you present to your immune system, somewhere in there there'll be a B cell that will recognize that, which will then proliferate and produce an antibody that recognizes that protein. But that's not by any means the whole story, as we'll see. It's quite a bit more complicated than that. So, if we look at the structure of an antibody bound to an antigen, so this is a Fab fragment, just the variable regions of the light and heavy chains, and it's been complex to a protein which is actually derived from the cell wall of the malaria parasite. And this is an actual structure, so the complex has been made and it's been crystallized, and the X-ray structure has been solved. And I've shown this here using one of the representations I showed you last time with sort of smoothed out van der Waals radii, but you can see here the antibody chain underneath, and you can see the different domains here. And what this picture shows you is essentially, it is like sort of a hand fitting into a glove or a lock fitting into a key. You can see that the antibody really does dock into that antigen very precisely. So the thing will just slot in, and the two surfaces are very complementary. Like one is an impression of the other. And of course, this makes the binding between the antigen and the antibody very tight, because there are lots of opportunities for the side chains in the antigen to interact with the side chains in the Fab fragment. And the more interactions there are that take place, the tighter that binding will be. And you want the binding to be tight because you want the recognition to be as good as possible. Now, at a molecular level, what are those interactions? What are the actual forces? First of all, they're non-covalent. So antigens and antibodies don't covalently bond to each other. There's no breakage and linkage of peptide chain. There's no disulfides formed or anything like that. But there's lots of other interactions that should be familiar to you in the base on from what you know about protein structures. And these could include hydrogen bonds, which can be, none of these are particularly strong, but there are many, many of them, so that's why the binding is so tight. So you can have hydrogen bonds, you can have van der Waals forces, which only really work when atoms are really quite close together, but they're very important in protein-protein interactions. You can have salt bridges. The salt bridges is essentially an electrostatic interaction between two amino acid side chains with opposite charge. So it might be, for example, an arginine or a lysine, which carries a positive charge being attracted to a glutamic acid or an aspartic acid, which carries a negative charge. And then there are hydrophobic interactions, and hydrophobic interactions are a little bit complicated to explain, but they take place between residues which don't like to associate with water, and so they quite like to associate with each other. So if you've got side chains which don't carry any charge, so they're non-polar, so things like valine, isoleucine, and so on, they, in the act of getting away from water, have to interact with other hydrophobic side chains, and that's called a hydrophobic interaction. It's not a bond as such, and the thermodynamics of this are to do with entropy, and they're quite complicated, and I don't want to go into them, but take it from me that all of these things are quite weak, but when you've got many hundreds of these interactions taking place in a protein, they're actually pretty strong. So it's a little bit like a sort of Velcro. When you've got two Velcro surfaces coming together, there's no physical linkage, there's no actual physical binding between them, and each individual interaction in that sort of hook and eye linkage is fairly weak, but you know, Velcro can make quite a strong contact because there's lots of interactions. Now, I made the point that with antibody molecules, the recognition part of the chain is not a linear part of the chain. So that was the point I was making going back here, that if you look at these regions, they're not actually adjacent on the peptide chain in a linear sense. They're quite widely separated, as is shown here. And exactly the same thing can be true about the antigen. So the region of the antigen, the antigen is the protein that's recognized by an antibody. The region of the antigen that's recognized is called the epitope. So the epitope is the particular part of the protein that a given antibody is recognizing. And again, these don't have to be linear on the peptide chain. They may represent different bits of the peptide chain which are brought together by the way that the protein folds up in physical space. Okay, so I've told you a bit about the fact that there's a lot of variation in the complementarity determining regions, and that gives antibodies a large part of their ability to recognize antigens. But I haven't addressed the question about where this variability comes from. So we've still got the problem that in order to encode these millions and billions of different sorts of antibodies that are circulating around the body, and the B-cell receptors and the T-cell receptors, we would need billions and billions of genes, similar in sequence but not quite identical in sequence. And that's just not on. The human genome is nowhere big enough for that. So we've still got the question, where does the diversity in antibody structure actually come from? I see that it's 10 to 11. Do people want to take a little break at this point before we get on to diversity? We'll take a 10-minute break. I'll get a coffee. You can get whatever you like. And then we can carry on afterwards. We'll start again at 11.